So today I will talk about biomarkers, both prognostic and predictive biomarkers, especially in response to immunotherapy. So my lab has been working since many years on the deep characterization of the tumor microenvironment, and we found some very interesting biomarkers that can predict the likelihood of response to treatment, the likelihood of having a tumor recurrence, who can predict patients with long-term survival. We have been mostly focusing on really characterizing the immune infiltrates within human tumors. So looking basically at all the immune cell subpopulations within human tumors. And we analyze what we call the immunome, that is the genes that are specific for every immune cell subpopulations. And so we analyzed many different cell subpopulations, immune cell subpopulations infiltrating the tumors. We found out that the adaptive immunity, and in particular the memory T cells, were essential to predict long-term survival. And more strikingly, what we found in the paper that we published in Science was that, in fact, the tumor progression and the tumor invasion, the tumor aggressiveness, all the classical parameters of tumor staging are in fact dependent on the host immune response and on the cytotoxic memory T cells that are present within a tumor, which of course change a lot the paradigm of how we see a cancer. And now that we have immunotherapies that can modulate these T cells that we have characterized, of course, this has a huge impact on the patients. Sure. Yeah, we have published both in New England Journal of Medicine or in Science or in Journal of Clinical Oncology papers around the importance of the adaptive immune cells, the Th1 markers like TBX21, chemokines, signatures with chemokines that were published. Of course, the memory T cells that we published initially back in the New England Journal of Medicine. And we had these studies looking at either early stage patients, and even at early stage patients, these immune cells are very important to predict patients at high risk of tumor recurrence, even though they have an early stage small tumor. And in the opposite, or likewise, for patients with very late stage metastases, like brain metastases, when analyzing these cells, these immune cells, with an assay that is called the Immunoscore assay. This is a standardized immune assay that we developed. This has a very strong prognostic value in stratifying patients with long-term survival in brain metastases. And that was published this year. We actually wrote a review published in Immunity about this topic and there is a strong relationship between some aspect of the immune response to cancer and some of those immune markers that have both a prognostic value and a predictive value of response to treatment, and in particular of response to immunotherapy. This has to do with the fact that the patients that have long-term survival, that are protected against tumor recurrence, needs to generate a T cell adaptive immunity. They need to generate a memory T cell response. And to generate this type of response, there is the need to have a Th1 type of response with interferon gamma, which upregulates these checkpoint receptors. And so these markers like PD-L1, the expression of PD-L1, has been shown to be a predictive biomarker of response. But it's not only PD-L1, it's in fact the adaptive immunity, the Th1 response, the densities of the cytotoxic T cells that goes along with PD-L1 expression that has been nicely associated with response and prediction of response to immunotherapies like anti-PD-1 treatment. So there is a paper published by a group from Tony Ribas, published in Nature, showing that the densities of the CD8 T cells at the invasive margin of the tumor in particular are very powerful in predicting the responder, the patients responding to anti-PD-1. Of course, Immunoscore is the most advanced because it's a standardized assay, and it's now undergoing a massive international validation with 23 centers across the globe, all testing Immunoscore with standardized operating procedures, with the same standardized assay on their patients. So in all the countries from Asia to US, to Canada and Europe. And so we expect the results now to come very soon. So this very large consortium, international consortium, that is currently testing in the same way an immune biomarker, I think, will make things change into how cancer patients are classified, because today worldwide there is absolutely no immune markers that are used in the clinic to tell something about the immune response of a cancer patient. There is none. Nothing is done in routine. There are some that are done in research or in some clinical trials, but nothing in routine yet. And so we hope that this will open the door basically to immune biomarkers in the field of cancer.